Hello, everyone, and thank you for coming out to the iGEM Health Equity Symposium hosted this morning by the University of Michigan iGEM team. This is the first time we are hosting this event, and we are very excited to be doing so. We're hoping that this will kind of create a forum for iGEM teams all working on health equity related projects to kind of share ideas, share resources, et cetera, and hopefully forge collaboration. As well as kind of create a community to encourage other iGEM teams to pursue projects related to health equity. So here's an outline of today's event. So we'll begin with some opening remarks. Then we'll jump into team presentations where each of you will discuss kind of how your projects this year relate to health equity. We'll go into a brief break, and then we'll have some time for you all to kind of ask each other questions and have some more in-depth conversations about potential collaborations for those of you who have similar projects. Then finally, we'll have some closing remarks. And here's an outline of the order in which the teams present today will present their projects. And once again, we'd just like to give everybody a warm welcome and our thanks for coming to today's event. And before we just get too deep into it, we will first introduce ourselves. So my name is Emily Wallace, and I'm one of the current co-presidents of the University of Michigan iGEM team, and I'll be one of today's moderators. And later today, you'll hear from Sophia Tessick, who is the other co-president of our team this year. So first, we'll begin with team presentations, and we'll begin with the University of Michigan's, which is our team. And with that, we'll get started. So this year, the University of Michigan's iGEM project is focused around trying to mitigate cardiovascular health inequity by empowering personalized medicine. So beginning with the high-level problem. So as many of you maybe know, thrombosis is a common clinical condition. Sorry, Sophia, one sec. Lights went off. Thrombosis is a common clinical condition in which patients experience clotting of blood within their veins and arteries, and this contributes to other clinical problems like stroke or pulmonary embolism. It affects about 1 million people worldwide and is a very common problem. And its relevance to kind of health inequity is that African-American patients experience thromboembolism at a 30 to 60% higher rate than the general population. So clearly there's kind of disparate outcomes in these populations. And so our team is interested in developing cost-effective solutions in order to kind of make these outcomes more equal and hopefully prevent certain populations, but particularly African Americans, from experiencing these events at disparate rates. So to dive a bit more into our problem mechanism that we're going to be focusing on throughout today's presentation and that we're focusing on in our project this year, is based around a protein known as PAR4. So this is a receptor that sits inside the membrane of platelets, and it's partially responsible for platelet activation. So this receptor will sense kind of environmental factors, and then in response to these, it will cause thromboembolism or clotting of the blood. So in PAR4, there's kind of a wild type or a majority, at least in the general population, version of this gene, in which there's an alanine amino acid at one point in the gene. And this kind of causes the normal mechanisms that we traditionally think of when we talk about PAR4, response to thrombin, which is a common signaling molecule, and how that leads to thrombosis. So Dr. Michael Hollenstadt at the University of Michigan recently identified an SNP, or a single nucleotide polymorphism in the PAR4 protein, in which this alanine amino acid is converted into a threonine. And this singular kind of mutation causes the PAR4 protein to be more sensitive to thrombin. So the same quantities of thrombin will cause increased rates of thromboembolism, which kind of contributes to patients experiencing blood clotting more frequently than they should. And to kind of circle back around the health equity focus of this, this SNP is about 30 to 60% more prevalent in African American populations than it is in populations of European descent. So more specifically, the threonine, so that alternate variant is found in the minority of Caucasian patients, about like less than 19%. However, it's actually the majority allele in patients who are African American, composing about 63% of this population. So kind of these disparate effects of these different alleles on the patient populations is likely contributing to differences in treatment outcomes for both of these groups. So to kind of add on to, not only does our SNP cause increased rate of thromboembolism and increased risk of these kinds of cardiovascular events, but it also has implications for antiplatelet therapy efficacy. So antiplatelet therapy is the kind of drug that somebody is prescribed in response to a clotting event, so in response to thromboembolism, in order to prevent clotting. So the most commonly prescribed drug kind of in this realm is a combination of aspirin and clopidogrel. So this is the standard of care. It's covered by most insurances, and it's just overall the most common. However, when patients have this alternate threonine variant of the PAR4 protein, this combination drug becomes less effective. In contrast, there are two other options for antiplatelet therapy known as ticagrelor and heparin, and these are much less commonly prescribed for a wide variety of reasons. However, they do maintain efficacy in patients with the threonine variant. So when we're kind of thinking of a solution to figure out how we can kind of level the baseline for antiplatelet therapy prescriptions, we're looking for a solution that is economical, so it can be affordable to most patients. It's effective, it's accessible, and has low barriers to entry, and also is specific and kind of does the job that we expect it to do. So our kind of like high level idea of our solution is to develop a point of care test for this alternative threonine allele, such that doctors can make more informed decisions about antiplatelet therapy prescriptions. So for instance, kind of as I described in the previous slide, a doctor can choose to prescribe an alternate therapy for patients that have this variant that might maintain efficacy better. So just kind of reiterate how the situation or how our test might be implemented. So of any patient experiencing a clotting event, thromboembolism, et cetera, requires antiplatelet medication. So the way that this is typically done is from this first initial step, a doctor will jump all the way to the end on the top right, where they will immediately prescribe aspirin and clopidogrel without any informed decision-making. The alternative method of decision that we're proposing is that every patient is tested for this alternative threonine allele in the PAR4 protein. If the patient is negative for the threonine allele, then they can be prescribed the standard of care, aspirin and clopidogrel, without fear that this medication might be less effective for them. However, if the patient is positive for the threonine allele, the physician knows that the clopidogrel and aspirin combination is likely less effective for the patient, and therefore they can be prescribed one of these alternative medications like ticagrelor or heparin that maintain their efficacy. So kind of an overview of how our solution might be actually implemented in a clinical setting is that a healthcare professional would collect a patient sample. So this might be something like blood, saliva, et cetera. And genomic DNA would be extracted from the sample. Next, the DNA would be amplified in order to enable detection of our specific genes. We would be amplifying the F2RL3 gene of interest. And we hope to do this through a mechanism called loop-mediated isothermal amplification, or LAMP. And we're hoping to use this as an alternative to standard amplification protocols like PCR, because it requires one study temperature, which enables clinical settings that don't have access to sophisticated equipment like a thermocycler to be able to still perform our test. Then once we have our gene amplified, we hope to use a sync and probe competitive binding system of fluorescent probes in order to bind to DNA with the SNP or not, and provide a fluorescent signal that correlates to whether the SNP is present or not. So just kind of wrapping up, how this solves the problem, by kind of pitching or by developing the solution that we describe, We feel that we can substantially kind of reduce costs of the kind of genetic testing that we're talking about, and enabling physicians to provide a more effective means of care for their patients, specifically when it comes to thromboembolism, and empowering these doctors to be able to make more efficacious prescription decisions for all of their patients, and ensuring that all of their patients are able to receive a drug that works for them. So kind of ideally, once we have all of this development complete, we're hoping to develop kind of a final end product of an end-to-end test that can be, sent to a clinical setting and can be entirely used in itself to detect for our specific SNP of interest. So kind of what would be contained in this kit would be everything from sample patient collection tubes. Our team has also developed a one-step DNA extraction buffer that can be used in a low resource setting and doesn't require multiple steps in order to effectively extract the DNA. We're also going to include our LAMP primers that our team has custom designed for the F2RL3 gene in combination with LAMP reagents needed to perform that entire amplification process. Additionally, it will come with the fluorescent probes we will use in order to detect the SNP or not in our LAMP product. And then lastly, our team is also developing a low-cost fluorimeter in order to detect differential fluorescence levels between SNP and non-SNP samples. And this is kind of just an addition that we've added on because we've recognized that fluorescence equipment is not necessarily inexpensive and may be inaccessible to healthcare settings interested in our test. And with that, we're happy to take any questions that you all have today. Thank you so much for listening.
Hello.
Hi. Feel free to, shut your
Hi.
question out.
Yes. So for your proof of concept experiments, are you guys using blood, or are you guys using simulated blood, or how are you planning to go about it?
Yeah, so for our initial testing, we've just been using DNA oligos of our gene of interest. So we just have an oligo ordered just from Twist that has the SNP and then one that does not. So we've been using this just to confirm that our LAMP primers work, our LAMP is happening successfully, and we're able to amplify our gene, and that our probe sequences work well and are able to distinguish between an SNP and non-SNP sample. And then we've been working on actually amplifying the gene out of a mammalian cell sample just with a cell line. We've been using HEC293 T cells because obviously all mammalian cells or all human cells have this gene. And then the next step was intended to be blood. So we have a collaboration with a PI here at the University of Michigan that is the one who kind of discovered this variant to begin with. So we were originally going to get genotype blood samples from him, and then we've run out of time a little bit and haven't quite gotten to that step, but we're hoping to maybe continue that in a future year.
Awesome.
Max, did you have a question?
Yeah. So how far have we gotten on developing the fluorimeter? Because it seems quite an ambitious plan to develop that yourselves.
Yeah, so we have a whole kind of sub team working on that. And we've been working on that since January. So it's kind of been a year-long endeavor. We've had a lot of consultations with some different faculty at our university. Luckily, a lot of our team studies biomedical engineering, so is pretty familiar with circuitry and whatnot. So we have our prototype fully developed, and we're currently working on, we have the CAD design, and we're currently working on 3D printing the fluorimeter. And then we're actually testing next week to see whether it's working or not. So yeah, but we have our design fully complete and everything. We're also hoping to make that design kind of openly accessible on our wiki, so that any other team that's interested in making a fluorimeter in the future can do so.
Cool, thank you.
Excellent. Okay, well, with that, I think we will let the next team present. So I believe that team is PathoGlow, if we have anybody here from that team. Is anybody here from PathoGlow, or no? We can move on if not. Okay, is anybody from Oasis here?
Hi, that would be me.
Okay, excellent. Feel free to, I don't know if you're just going to talk or you have screen sharing abilities as well if you're interested in presenting something.
Yes, we can present. Just give me a second. I can figure this out. So can you guys see my screen? All right, I'm guessing that you can. Am I audible as well? Okay,
Yep,
cool.
we can see
So,
it already.
okay. Hello, everyone. I am Vibhavari. I'm part of the ICER TVM iGEM team. And we are working on a project that's called OASIS, which is a FRET-based microfluidic device that can be used to diagnose MDD or major depressive disorder. So I'll get into it. Okay. Yes. So obviously, we're all familiar with what depression is. It is a symptom-based, mood-based disorder. So it is characterized by, again, symptoms like loss of interest in daily activities or not having the energy to do anything or fatigue, tiredness, all of that. But when these symptoms persist for longer, so they can show up as physical symptoms as well. So they can cause insomnia, they can cause appetite changes, all of that. And when these symptoms persist for like 2 weeks, 4 weeks, longer than that, it's a chronic illness and it's called major depressive disorder. A large chunk of the population is depressed, and India has a very high rate of MDD cases. So does Kerala. Kerala is where we are from, and which is why we started working on this project to begin with. Right. Why is depression diagnosis a necessity? One thing is that I will just move on to why we thought we should choose this topic. So one thing is that the current methods for diagnosis, they mainly rely on self-report questionnaires. So you're given a questionnaire and you're given your ratings and rate them from this to this. And based on that, subjective evaluation of these symptoms happen. And if you say you have these, then the doctor has to diagnose you with these symptoms. And then that is usually followed by like a clinical interview where they assess your body language, non-verbal cues, all of that. But the problem with this is that it can introduce subjectivity. It can introduce a personal bias. So it can change from a psychiatrist to another psychiatrist. And it's overall not the most reliable way. So what we are trying to do is find an objective aid to this diagnosis process. So currently in market, there is no reliable quantitative tests. There are a couple of quantitative tests that I will go into later, and how they work. They work on a single biomarker. So they quantify that. And usually that does not tell you a lot about whether or not this person has high risk for depression because it is not specific at all. Right. And so one of the other reasons is that, especially around mental illnesses, there's a lot of stigma. And it's very easy for people to say that, for people to invalidate your experiences, even though it is a disease. Right. And so a solid objective test to this should reduce the social stigma around mental illnesses. So what our project is, it is a microfluidic device, and we are working with 5 different biomarkers. We are trying to quantify all of these 5 different biomarkers using 2 quantification systems. And what this would mean is a sample, a human blood sample, and our chip can analyze that, and we will finally be analyzing it. It will produce fluorescence. This is a FRET-based system, and this fluorescence can be detected by our hardware system as well. So what we've realized is that it's not just a diagnostic tool. It can also be used to track antidepressant treatment. So say a person starts on medication with antidepressants to see whether or not it is working properly, to see whether or not it is, because antidepressants work very different. Depression is a very heterogeneous disease, and it shows up very, very differently in different people. And so antidepressants, the kind that is prescribed to people, all of those also don't work. Each of them are not very specific. And so what we thought was that we can use this tool itself to sort of use it for the checking how the treatment is going. And so I think we have another teammate here. She can proceed with how.
Hello, I'm
Rashmi.
Roshida. Am I audible now? Okay. So continuing from what Vibhati said, we have gone through extensive literature review and chosen 5 different putative biomarkers. We call them putative because they are not established biomarkers yet. So we have chosen 2 microRNAs, which are microRNA 132 and 124. And then we have serotonin and cortisol, both of them known hormones, and serotonin, which also acts as a neurotransmitter. And we have GS-alpha protein, which is a receptor-associated protein. So microRNAs. So the reason we have chosen 5 biomarkers are to so that we can reduce the bias which is caused by looking at just one kind of biomarker. So microRNAs, they work at a more transcriptional level, whereas your serotonin and cortisol, that would look at the more translatory mechanisms. And whereas the GS-alpha is, as I mentioned, is a cell receptor associated protein, so it would be looking at cell-to-cell communications. So we are really getting into the physiological basis for MDD and trying to look at it from more overview as a more broad view, so that we can avoid any kind of bias that might arise. And so moving on to our quantifiers, we use 2 different kinds of quantifiers to measure the concentrations. So the first kind of quantifier is called FRET optasensors. Both our quantifiers work on the phenomenon of FRET, or Foster resonance energy transfer. So this is basically a method of non-radiative energy transfer between 2 molecules, a fluorophore and a quencher. So we are using ultrasensors to quantify cortisol, serotonin, and GS-alpha protein. So each of them would have specific aptamers, and we all know how aptamers work. They are very specific nucleotide sequences that bind to your target molecule. So what we have done is we have tagged a fluorophore molecule to our aptamer, and attached the small part of that aptamer as a complementary DNA strand, which will have a quencher molecule which is tagged to the end, to the complementary end, which binds to your aptamer. And so whatever fluorescence is emitted by the fluorophore is now absorbed by the quencher, and initially, so this aptamer cDNA complex would not show any fluorescence. However, in the presence of your biomarker, or here if we have shown cortisol as an example, in the presence of cortisol, the cDNA gets displaced due to more affinity towards cortisol, and a new biomarker aptamer complex is formed. So this would mean that the fluorophore quencher energy transfer is now inhibited, and this would result in emission of fluorescence. And this fluorescence is analyzed for measuring the concentration of cortisol in your biomarker. This is the same way we plan on measuring both serotonin and GS-alpha proteins as well. Moving on to microRNAs, we have tried to engineer something called magnetic nanoprobes. So nanoprobes are nothing but a double loop stem structure made out of oligonucleotides. These would again have as part of their stem, they'll have a fluorophore and a quencher molecule, which will inhibit any fluorescence from being emitted. And in the presence of microRNAs, specific. So again, these are also specific to 124 and 132. They are specific probes. So in the presence of your microRNA, the loop part of the structures, as you can see, the round part of it opens up, which will again pull apart your fluorophore and quencher. This would again result in emission of fluorescence, which is quantified to figure out the levels of microRNAs. And since the concentration of microRNAs are very less in blood compared to the other biomarkers that we have chosen, what we have done is we have tried to attach all these nanoprobes onto a magnetic nanoparticle, so that it gives a more accurate and a more reading from a smaller amount of sample, and more collected reading. And now that we have both our quantifiers and biomarkers in place, we are trying to scale down all of these onto a microfluidic chip, so that it can be, integrated into the real world diagnostics techniques as of now. So this is the preliminary design that we have, and on the left side where we will add your blood sample. This will get separated into 5 different channels. Each of these channels will contain your quantifier. And on the right side of it, it gives basically fluorescence readouts. And we are also trying to develop an associated sensor which can analyze these fluorescence readouts, and all the results will be again processed on a microchip, which will give us the result, which would be a high risk or low risk for depression, basically.
Right. And so obviously for the future of this, now that we are getting the considerations of biomarkers, what do we do with them? We obviously need to find a threshold for it. So the problem is that there isn't enough data right now, because all of the data has been for smaller clinical trials, smaller studies, not really generalized for the entire population, right? So the first step would be to find the threshold for this, to sort of build the data, right? And so once that we have that data, we can set the threshold values. So we can use the chip for that. And then we can move on to permissions and everything that is required to bring this chip into the trials, right? To conduct trials. And so this is the basic overview of our project. We would be happy to take any questions.
I have a quick question about, we have a very similar, or I guess it's different, but kind of a similar mechanism with using the fluorophore quencher system as a means to detect various things. I'm curious if you can go back to that slide quickly. Yeah. So is your quencher 100% complementary to your fluorophore sequence, or did you guys have to do any engineering with this system in order for it to work the way that you were anticipating?
So we have ourselves developed the complementary DNA strands. We have ourselves engineered it, and we are trying. The way we have gone about it is we have tried different combinations of cDNA of varying levels of complementarity, and we ran experiments for all of them to figure out which one works best.
I see. And so by varying degrees of complementary, did you just induce various point mutations, or did you play with like multiple nucleotides not being complementary? Like, I guess what was the range that you tested?
So we did point mutations, and so I think so for one of them we did point mutations, and for the other we had multiple nucleotide variations, I think. Some of the sequences are shorter. Not all of them are of the same length. Some of them are shorter as well.
Okay. And then which, I'm just out of curiosity, which one ended up working the best for you guys? We're having kind of a similar type of engineering to do with our fluorophore quencher kind of pair to see what is most optimal. So I'm curious if our results were similar at all.
So we've actually ran the experiments, but we're yet to analyze all of them. So,
oh, okay. Understandable. No worries. Okay, sounds good.
Anything else?
I do have a quick question. So I really like your guys' idea, of taking all of these 5 biomarkers at once. And you mentioned that not all of them are, I forget the wording you used, but, verified in literature. These are like things that have been hypothesized. So I was just wondering where you collected the 5. Did they come from all the same source, or you really chose to focus on things that, you wanted a protein, you wanted microRNAs? Like, how did you come up with these 5 specifically?
So obviously the most obvious, so we look through the kind of biomarkers that are most relevant to MDD already. And so we came across serotonin, dopamine, all of those, the neurotransmitters, the very obvious ones. And then we realized that we wanted this to be like sort of a blood test, right? So we realized that dopamine as a molecule is not present directly in blood. Whatever relation that lower levels of dopamine have with with MDD is all in brain or cerebrospinal fluid, CSF. So it started there. It started with the most obvious ones, and then we reviewed literature. We sort of wanted to, like she mentioned before, sort of wanted to look at it from different levels. And so we went with microRNAs, which are just starting to come up in diagnostics. And the quantification of microRNAs is also like new and blooming. And then we have our GS alpha protein, which is a pretty novel one. So we have sort of a mixture of biomarkers over here.
We have also talked to professionals across fields and had them validated by different people who have been working in diagnostics, in psychiatry, in psychology, etc.
Can I ask a quick question? You might have said it, but I didn't quite catch it. If that's the case, how are you purifying the microRNA from the blood?
Oh, so our microRNAs were directly, we actually bought the, what are they called? The synthetic microRNAs, yes.
Yeah, I'm thinking in the
Oligo
future.
sequence.
In the future.
Yeah, what's
right.
the plan down the road?
Right. So it's for purification, we've only thought about for the protein itself. For the microRNAs, we are running experiments to see how specific our quantifier is to the microRNAs. So as of yet, we haven't thought of the purification methods for microRNAs.
We'll, as in there are methods for RNA purification, but we have to figure out how to integrate it all on a microfluidic set. We are still working on that.
Thank you.
Right. Thank you so much. So are we ready to move on to the next team if there are no other questions?
That sounds great. Thank you so much.
Awesome. So the next team we have, it should be Natriotech. Do we have any representative from there? Oh, perfect.
Yes. Can I just share the screen?
Yes, you
assume?
should have option too.
Yeah. Okay. Is this fine? Yeah. Okay.
Right.
Sindre,
You can hear
take it
me?
away.
You can hear me? Great.
Yep.
Yeah, so we are then Meteorotech. We're representing the University of Oslo here in Norway. And our project is regarding the diagnosis of endometriosis. Endometriosis is a disease characterized by the inner lining of the uterus, which invades the exterior of the uterus and can even invade further out into the pelvic area. And this invasion results in growth of endometrial tissue on the outside of the uterus, and the consequence of this are symptoms ranging from infertility, pain during sex, heavy and painful periods, painful bowel motion and urination, fatigue, and bloating, just to name a few. And the severity of the symptoms will increase with disease progression through the different stages. So why is there a need to diagnose endometriosis? First of all, it's estimated to affect 1 in 10 women worldwide, totaling about 190 million affected. And even though there are so many affected, the mean diagnostic delay is still around 7 years worldwide. Unmute that, Mats.
There you go. It's nice having actually being able to hear me. So what do we actually achieve when we diagnose the disease earlier? As we said, the symptoms do progress after the longer the symptoms get worse the longer the disease progresses. And this is represented by the endometriosis stages, which range from 1 to 4. And as you can see, the endometriosis kind of invasion is quite drastic when you get to the later stages. But it is possible to at least slow the progression if the patient goes on birth control. And that means that if you're able to get an earlier diagnosis, you can therefore prevent at least the development of the very late stages of endometriosis, as well as it might remove the need for laparoscopy, which is quite an invasive surgery and is currently the only way to diagnose endometriosis. So the goal of Meteorotech is to create an accessible and improved method for early diagnosis of endometriosis. So our plan for how we will do this is to extract microRNA. We're still trying to develop, at least look at this, but it's quite difficult, as the previous team also knows. But we need to extract the microRNA, and after we extract it, we're going to amplify it with micro RPA, and then we get a double-stranded DNA product. And with this double-stranded DNA product, we are using a dual Cas DNA complex to bind. And then these two proteins, one of which can associate with the chromophore that makes it glow, and the other one is associating with the lateral flow assay to make this test strip red. So yeah, our project, at least specifically the project, is to create a diagnostic method for endometriosis that detects upregulated microRNA from blood samples using a dead Cas12 system. Specifically, we want to use a panel of microRNAs to do this. So the principle of detection is that these two dead Cas12F proteins, they do not have any, they won't destroy the DNA because they're dead, but they are able to bind to the DNA. And then the two different, one of them is going to bind to a chromophore, and the second one is going to bind to the lateral flow assays. Yeah.
Okay, so for what we've done in the lab so far, thus far we have mostly just transformed E. coli cells, specifically BL21 cells, with the expression vector containing each of these dead Cas proteins, one of which is tagged with a his tag, the other with a strep tag. These will be used in both the purification and detection. These have been induced at appropriate conditions, and over time we have tested this. And Mats, if you could switch to the next slide. Yeah, we have induced these at appropriate conditions and then tested various conditions, since, as you can see, our first conditions, 37 degrees overnight, didn't work that well, with most of the protein, the largest block you can see in the insoluble or insol column, being collected in the fraction that is harder to work with during purification. So when we tested these various conditions, we found that the room temperature overnight, as well as the the room temperature overnight had the most optimum expression conditions, though the amount of inducer was also changed to try and find just the right balance for expression. Now we have come a bit further than this. We are done with the actual purification as well as the production of our guide RNAs, and we are about to try and test if our dead Cas system is capable of binding to a designer target double-stranded DNA molecule. Further than this, we hope to prove that at least we can detect a double-stranded target with this system, and that the system holds promise for adaptation to other diseases as well, if it works out. We had hoped to be able to work on the actual test chip, but as you all probably can feel on your own project, that there isn't that much time left.
Yeah, so quickly, this is the team, then the Meteorotech team. And we also want to give a brief thank you to our supervisors, of course. And if there are any questions, please come with them. If not, then we are done.
Excellent. Thank you so much. Do we have anybody here? Oh, sorry. Did you have a question?
No, I'm sorry.
Oh, no worries, no worries. I just wanted to make sure. Okay, great. Do we have anybody here from Oxford? I believe Erica, you were representing that team.
Yeah, right. So the thing is, we haven't done our presentation slides yet. So I judged the best way for me to present it now, since the preparation time is really short, is to just show our promotion video to all of you, and then ask receive any question if there is any.
Sure, that sounds great.
Thank you. I'm gonna share my screen. Can you guys see it?
Yep.
Okay, great. I'm gonna play it now.
There is a contamination crisis. This is the problem we at Back to Glow are determined to resolve. 1 in 3 people worldwide do not have access to clean water. And you would be mistaken for thinking all dirty water looks like this. Data from the JMP shows how widespread the problem is. The least developed countries, war zones, and regions affected by natural disasters are the most affected areas. Another major issue faced by organizations such as the JMP is the lack of clear data about this issue. Sporadic and varied testing leads to inconsistent data and poor understanding of the problems faced. In emergency situations or conditions of extreme poverty, water supplies are often cut short or inaccessible. Houses may be up to 30 minutes walking distance away from their closest water source. These range from piped water sources to wells and boreholes. It is important to test the water both at the source and at household level. Current methods of testing involve collecting water from both locations and then transporting the sample to a lab. Once in the lab, the water samples are cultured on plates and incubated for 24 to 48 hours. Quantifying the contamination then requires counting the number of colonies that have grown. Our solution is much faster, more specific, and easier to read. We have devised a biosensor that binds to specific strains and emits a luminescent signal within minutes. This removes the requirement for a lab or trained professionals. Testing can happen directly at the source and at household level. Back to Glow promises a revolution in humanity's ability to access clean, uncontaminated water with rapid and accessible testing. Collection of quantitative data from across the globe allows for more effective analysis of water contamination. With our technology, we will be one step closer to achieving the United Nations Sustainable Development Goals. We want to give everyone the opportunity to drink uncontaminated water. Back to Glow, shining a light on the water contamination crisis.
Okay, so that's it. Any questions? I mean, I do have something to add on to our presentation video to make, like to connect with the theme of this seminar. First of all, we participate in this seminar, healthcare equality seminar, because we feel like water contamination, especially in those regions where you don't really have good access to clean water sources, and you don't have access to good water tests. These feces in the water can very rapidly spread any bacteria or any other pathogens in the water and cause risk to the local people's health issues. And secondly, we are focusing only on bringing these rapid accessible tests to the poorer regions or regions where like an accident a disaster just happened, like Libya, for example. Right now they're facing very severe flooding issues in their own country. And so our goal is to suit the best to WHO's REASSURE standards, which is on diagnostics, making sure that these diagnostics are, first of all, accessible, and second of all, user-friendly to low resource regions. And I want to make sure that we are all on the same line of what does user-friendly mean and what does accessibility mean. I mean, these words sound like very broad, but bring it down to the diagnostic level. We can separate accessibility, for example, to first, one, portability, right? You need to bring the test around, because you might not like to the homes of the local people, to the water sources, and both of these regions will need you to like get off your truck and maybe keep it with you. And when you're doing the test, you might not have accessible electricity sources. So this is what portability means. And then secondly, accessibility also involves transportation, easy transportation. Well, along the way, you have to make sure that your diagnostic test is stable, and it can be stored in the truck for a long time in order to get delivered to the regions that needs them. And thirdly, of course, affordability, right? We have to lower down the price as much as possible. So whatever we're using as our components need to be able to be massively produced. So this is where synthetic biology can help us really a lot. Our tech is based on proteins only, and this synthetic protein. So, I mean, you can easily produce a lot of protein if you have your bacteria cultured and have them secreted, for example. And user-friendliness, right? You have to make sure that your steps are not too complicated, because the people that are actually using the test, especially in those resource-limited regions, they might not be very well trained. And especially if you're making a home test, it's very important. Also, you need to make sure that the sample collection is not too complicated. I'm sure like one group mentioned the process of collecting mRNA and extracting mRNA from the patient sample. I think this is a very good thing to consider when you're designing your diagnostics. If you're making it too hard to actually deliver this very first step of making the sample, then I mean, how can you do that like with patients directly? And yeah, that's basically it. And of course, like on top of that, a good diagnostics need to have good sensibility, good specificity, and a good limit of detection that is on the level that we needed for different pathogen sources, or biomarker sources. So all of that putting together, it makes making a diagnostic tool for low resource settings exceedingly difficult. And this is what we're targeting on. We want to focus just on making it more friendly, making it more user-friendly, making it more accessible to the low resource regions. So yeah, that's our project.
Thank you. So a question that I had, I know people don't typically like dive into specifics in their promo videos. So I was just hoping, could you talk about specifically what you were aiming to detect? You mentioned like multiple strains, I think, in the video. And I know like water isn't just one thing. So I was just wondering if you could talk about that.
Yeah, of course. So actually, maybe I'll share my screen again. See. Yeah, so this here specifically is our system. So can you see my mouse, by the way? I'm not sure. Okay, good. So this thing my mouse is pointing, it's a well, you can call it a biomarker or just a surface protein on a specific type of pathogen. Any strain, E. coli, for example, this is what we're doing. And these two are the synthetic proteins that we're designing. So this bit right here, it is the binding region that we basically just pull out of bacteriocin. So bacteriocin is a bacteria-derived pathogen, bacteria-derived toxin that targets very specific strains of bacteria. And they bind to the surface proteins, make a hole, get into the bacteria, and then do their toxin work. And that's what they do in nature. And then we just pull this very specific sequence out that binds to that basically recognize the surface protein on the strains that they recognize, and we put it on this bit of our protein. And so before, they're like this, right? That big protein is like this. So there are interaction between these two alpha helices. And then upon binding, it opens up, allowing the second protein, the second synthetic protein to come in. And these two gray parts here, both are parts of a luciferase. So the opening up of this dark blue protein allows the binding of the light blue protein to the dark blue protein, which allows the binding of these two gray bits together to form a functional luciferase and gives off a signal. So this is the basic mechanism of our system.
So you're just working initially to detect, E. coli presence in the water? Is that your, starting point?
We're also we're making two strains detection right now.
Right.
E. coli is one of them. It's easier to start, and then the other one is Klebsiella.
Yeah. I was just curious. Thank you. Do you have any other questions? All right. If not, then thank you, Erica. And do we have any representatives from Tongji, China?
Yeah. So I will share my screen. Okay, so thank you so much for inviting us. And I'll start our presentation. So is our slides shown normally? Okay, so then I'll start. Okay, so thank you for having us here, and today is our National Day. We're Tongji China from the College Life of Science and Technology, Tongji University, PR China. And our project is called PLINK. It's a high-precision quantitative detection system based on highly conductive protein nanowires. We have to develop a highly scalable system that combines electronic circuits and biomolecules to provide a sensitive and convenient tool for the detection of antibodies. And so first, a brief background of our project. So our project aims to improve one of the core diagnostic steps in the medical field, is antibody detection. Antibody detection is an important method to determine the presence of some specific pathogens or disease markers within human or environmental samples. We approach this problem from a synthetic biology angle and we want to give an innovative solution to this problem. Various antibody detection techniques are widely used today, such as direct, indirect, double antibody sandwich, ELISA, etc. Facing many challenges in practical applications, such as difficulties in quantitative detection and cumbersome and complex operation, and also harsh condition limitations of use. So we have encountered our key problem is that we want to build an easy system that can detect antibody in situ. So we've got our inspiration from Geobacter metallireducens, which has a highly electrically conductive pili. It's a type 4 pills, which has the highest conductivity of any biological material that has been discovered, which is very fancy. And it is generally believed that its high conductivity is related to the distribution of aromatic amino acids in the protein. But Geobacter metallireducens is a gram-negative and strictly anaerobic microorganism isolated from aqueous sediment with harsh growing conditions and immature and unstable culture techniques. Therefore, it is important to express the pili of Geobacter metallireducens in more commonly used traces organisms. So now we found Vibrio reagents. It is to our knowledge the non-pathogenic bacterial species with the highest reproduction rate ever known. We designed the target sequence based on the Geobacter metallireducens. Both Geobacter metallireducens and the Vibrio reagents possesses type 4 pillars that rely on a highly conserved assembly system, the type 4 pili assembly system. So we first accomplished sequence adjustment to correct heterologous assembly, and after that, we designed the model. We analyzed the peptide structure and made epitope tag modifications to the pili, which simply is just link the antigen to the pilus monomer. So in order to better determine that what we obtained from the Vibrio reagents is a heterologously expressed pili of Geobacter metallireducens, we used the CRISPR-I system to repress the genes of its own pili, which is non-conductive, and thus facilitating our direct observation of the expression and assembly of this conductive pili on its surface. And we designed 4 sgRNAs targeting the vicinity of the TSS, the transcription start site of the gene for the pili of Vibrio reagents itself, and selected DCAS9 as the repressor. To verify the efficiency of the CRISPR-I system, we constructed a reporter plasmid, which will illuminate if it works normally. Secondly, I'll briefly talk about our human practices and our social responsibility. We have gathered data of current prices of physical examination programs in different hospitals and third-party institutes in Shanghai, China. As are shown in the box plot, and compare the data with the local expenditure level provided by the National Bureau of Statistics, as are shown in the scatter plot. And at present, the community health service centers with the best medical testing facilities in the community are unable to undertake routine antibody testing programs in medical checkups. And these programs are usually entrusted to qualified companies with longer testing cycles and higher costs. While the main indicators of the routine health checkups of the residents and the door-to-door checkups of the family doctors in the community are still limited to some simple items like blood pressure and blood glucose. So, but antibody tests such as autoantibodies, anti-phospholipid antibodies, and immunoglobulin checks are commonly items used in health examinations. For various reasons, assisted or auxiliary diagnostic techniques have not yet been widely used here in health examinations. And what we hope is that auxiliary diagnostic techniques can promote the progress of health examinations in the direction of being faster and more accessible and cheaper. So antibody tests such as autoantibodies, anti-phospholipid antibodies, and immunoglobulin sets can be just be assayed by some subtle shifts in our system. We just need to change the epitope tag that is linked to our pills. And in our development, the industry and the academy has shown a lot of support. And also we've been to the communities. And here you can see is a neighborhood center. And I'd like to introduce that. The residents committee here is a mass self-governing organization at the community level. And here you can see this lady, Miss Xie, she is the president of such a committee. And she told me that the cooperation model between the resident committee and the offices of the people's government are very close. And we've got to know about the health examination services in the community level has been free for the residents. And this service is provided by the Community Health Service Center. And we have seen that with the effort of many parties, the health welfare of residents have been promoted and safeguarded. And the residents maintain expectations for more universal and convenient health checks. Also, we've been doing service in the communities nearby. So here I'd like to draw our conclusion. We finally conclude that the social solution to The problem is, firstly, improving the status of auxiliary diagnostic method in healthcare, and secondly, empowering the communities to promote the equality in health and well-being. Okay, that's all, and thank you very much. Any questions?
Right. If we don't have any questions, then do we have a representative from William and Mary here?
Hi, yes.
Thank you.
Okay, can you see my screen? Okay. Okay, so hi, my name is Libby. I'm on the William Mary iGEM team. And this is our presentation about the connection between health equity and soil symbio. So what is the problem? The current state of soil health, it is somewhat in a crisis with the advent of high-scale agriculture, increased use of pesticides worldwide. Soil health has really been degrading in nearly every region of the world. So soil health is defined by the USDA as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Soil health is important for ecosystem biodiversity, safe and productive agriculture, for maintaining the nutritional levels of crops, and for the prevention of soil pathogens. And some of the current major threats to soil health are pollutants, loss of biodiversity, depletion of nutrients, and instability. And some of the main threats in terms of contamination are petroleum, pesticides, urban waste, and chemical and biological warfare. So this is just a graph showing the increased use of pesticides in every single region in the past, or from 2000 to 2017. So the global average is 34.3% increased use of pesticides. This is a graph showing nitrogen levels across time. So the red line is nitrogen due to fertilizers, and the blue line is the amount of nitrogen that crops are able to remove. Ideally, these two would be equal, but as you can see, in many countries, the amount of nitrogen from fertilizers is pacing out the amount of nitrogen that crops are able to remove. So what is the connection to health equity? Since soil health is necessary for food access and agriculture, a lack of nutritional crops turns into an environmental justice issue and a health equity issue. So here are just some quotes here. The impacts of soil pollution continue to pose risks to the health of frontline communities, and healthy food access policies and racial justice go hand in hand. Besides food, there are also some issues with water runoff and soil health. So areas with poor soil health have increased chances of water contamination, as well as increased chances of being exposed to pathogens. Okay, so what are some potential solutions to these problems? There are a lot. This has been a concern globally for a long time now. So chemical treatments have been developed, including stabilization and chemical oxidation. Phytoremediation is also a popular technique. It's when people use plants to remove contaminants from the soil. There is also bioremediation, which is what our project is interested in this year. I'm sure most of you probably already know what bioremediation is, but it's basically enhancing this natural process that occurs in the soil where microbes degrade different contaminants, but either by adding nutrients, increasing amounts of native microorganisms, or adding engineered microorganisms to the environment. And there's a bunch more. So our project, as I kind of hinted at, we're interested in bioremediation. So our project this year is called Soilution. We're working on gathering foundational data for soil synthetic biology to be possible. There's a need for soil synthetic biology because the use of engineered microbes for bioremediation is a very promising solution to remove dangerous pollutants from soil. And this is especially true for soil environments because the soil matrix is super complex and you kind of have to remove pollutants in soil. In the soil, you can't really take it out and process soil effectively. So microbes is a way to treat soil in situ. So one of the issues that we've seen is a lack of fieldability in the field of soil synthetic biology. So a lot of people have been working on making constructs that are very successful in the lab, but once you test them in real world environments, they don't work. Either they'll die out or the construct isn't successful and does not degrade or do whatever you want it to do. And we are thinking that this is due to increased competition in the environment that they don't experience in the lab, as well as a lack of nutrients, because being in a nutrient depleted environment is very different from being in a flask with nutrient rich broth. Safety is also a large issue with soil, or safety is an issue with soil synthetic biology, but also with fieldability. It's hard to test fieldability if you don't have the proper safety measures in place to do so. So it's going to be difficult to test whether constructs work in large scale environments without knowing that it's going to be a safe experiment. So we're addressing this in multiple ways. On the safety side, we are working on genome integrating our gene of interest, which prevents horizontal gene transfer, which is a big concern with safety. We're also working on engineering phage, which would help deal with a scalability issue. So another issue with practicality is that it's very expensive to create the amount of microbes you would need to place them over, say, acres and acres of land. So if we were able to create a few phage that have our gene of interest and infect them in a specific point, and that's able to spread, that would be super helpful. And also because of host specificity, phage engineering keeps that safety aspect. So what are we actually doing in our project? We're collecting a lot of foundational data about how engineered microbes behave in soil. So we have an engineered Mycobacterium smegmatis strain that's engineered with a plasmid and it's genome integrated. And we're testing it in soil environments with differing concentrations of phage and with differing levels of competing microorganisms. And we also have a predictive model that is both informing our project and then we're going back and tweaking the model based on the data that we're getting. Okay, so here are my sources. That's all I have. Does anybody have any questions?
You maybe talk just super briefly a little bit more about what you're modeling, and like how much has it helped you so far? Anything like that.
Yeah, so we sort of have two aspects to it, but the one that's more far along is the spatial movement of bacteria in soil. So because soil symbio isn't really at the point where you can test how your engineered construct is going to move in like a large scale environment, we're able to model that through like fluid dynamic modeling to see how they would move. So for example, we weren't sure how spatially how far apart to take our measurements, and the model told us how far we think the bacteria should spread in a certain amount of time. And we're also taking into account metabolic processes, which we're hoping to integrate with that model, but we haven't gotten there yet.
Great, thank you. If we don't have any other questions. I think no. So do we have a representative from Gene Hackers here? Do we have anybody from Pathoglow who came later? Right, any other team that we missed somehow? All right, that actually might be all the teams then. So let me quickly pull up. So let me share my screen again. So we just quickly wanted to, before going into like time for discussion or more collaboration, this is an opportunity that a different iGEM team reached out to us with, specifically Iser Mahali, and we wanted to just offer it to you guys as well, as you know, sort of a last minute collaboration opportunity, in case you're looking to help another team and also include something in your human practices efforts. So they've also posted this on the iGEM Slack, but I'll give like a super brief overview. So the gist of what this team is trying to do is collect some manuscripts that basically briefly explain the scientific foundations of your projects. And so their intention is to collect many manuscripts from a whole like variety of teams, not just focused on healthcare inequality. But you know, pretty much anything that you guys have talked about today is definitely within the realm, but they're looking for pretty much any um, type of project. And so they want to publish them on their team wiki and then also distribute them to local high schools that they've partnered with in their area and also other universities. And their goal is to basically, you know, funnel or kindle this curiosity in high schoolers and undergraduates for you know science in general and also the realm of synthetic biology. And so if you guys are interested in basically helping them out, Team Michigan is also participating in this. Basically, they're looking for a short 2 to 3 page like manuscript or just scientific explanation of your project. And so we figured this would probably be kind of um, what's the word, like low effort from you guys because a lot of you have probably written up your wiki pages already, so something like the project description or other page could be, you know, cut down or just slightly modified to basically fulfill these guidelines. And I just wanted to quickly open up like the example that they sent to us. And so they just give a short project overview, some key objectives, and then explaining the technology behind their project. So, do we have any questions about that? We'll send this again in like follow-up email just in case you guys are interested in, you know, sending something like this. We'll include their email and you can send it directly to them, but just wanted to point that out. And so for the rest of the time here, we finished quite a bit early, which is always nice, but we figured that we could use this time for drop-in questions and collaboration. This can be pretty much anything. We didn't have a set format. So since there's you know, a small number of us here today, we can probably just stay in this main room, and then if needed, I can open breakout rooms so that different teams can talk to each other. But we were just envisioning, you know, maybe some discussion of you know, collaborations more on the HP side. What have you guys done that worked for you this year? What are some things that you are looking to collaborate on, maybe at the start of next season, sort of thing, since we all have a few iGEM teams here already. So I guess I'll just ask a few questions. How many teams here have done something with, you know, outreach to high school or secondary school students in their area? Team Michigan has done something, or any other like outreach, educational outreach. I'm just curious how you guys have gone about that, like some ideas that you had.
So the whole UK team, well, first of all, we only have seven like collegial teams in UK, so it's very easy for us to just connect with each other. So we have a WhatsApp group with all the outreach officers, etc. And for all the 7 teams, we make up a plan to produce some educational materials, including slides and texts, like little boxes of questions and just random definitions and stuff. So yeah, each team is assigned a topic for us. So Oxford iGEM did safety, biosecurity, and ethics in synthetic biology, and the other teams did, for example, like iGEM history and uses of synthetic biology in different fields, something like that. So yeah, so us compiled a whole booklet of things, of different topics and slides. We are not currently planning to do like actual outreach to the high schools, but some of the other teams, they want to do that in their local region, and they have all the access to different topics. So I think that's a very good way to like for us, for UK teams to collaborate. Yeah.
I can talk a little bit about what we've done. So it's actually been super helpful. The school, like William and Mary, has a lot of outreach to the community already. So we kind of worked with that. So like over the summer, there was a 2-week camp where middle schoolers and high schoolers came and did different activities. So we did a program with them where we had them like go out and collect soil, and we did some lab work with that, which I think they found pretty cool. And there's also been some other like community events where small children will come and learn about science. So we've presented at those sort of things.
That's great. Yeah, specifically Michigan, we went to a local middle school, so a little bit younger than high school. And we talked about, synthetic biology, just like a brief introduction, and they then gave, an overview of like what we were hoping to do with our project. We did that fairly early in the season. So I was just wondering if you guys did stuff like more generally to get kids interested, um, in synthetic biology, or like connected your project somehow. So it sounds like you guys did both, which is pretty great. And then I'm also curious, what have you guys like, we're always like looking for ideas for next year, and hopefully we can maybe give you guys some ideas and just like exchange some information. But what have you guys done like early on in the season to, like establish your like human practices connection with other iGEM teams? Because we, obviously tried to host this symposium. We, had a few meetings with teams to like engage in like a science-based discussion, so like some troubleshooting. But I was wondering if like any of you guys did something like more creative or anything like that that like really worked for you. If my question wasn't clear, I can also repeat it, but
So in India, we had something called an All Indian iGEM meet. I do not know if everybody does that. So which was a very good opportunity for one-on-one input, talking, interacting with other teams. We got collaboratively as well as input-wise. We basically all of the teams competing from India came together in an institute and presented our projects to each other. So that was a really good experience.
yeah, was that early on in the season when you guys had just basically outlined your project, or was it like midway so you actually
So
had,
it was
results? Or
midway. I'd say it was midway. It was around August,
okay. And you
August
felt
first
that
week.
that timeline was helpful?
Right, we could, so by then we also had like some amount of wet lab data to show, so we could actually show people and get more viable inputs for troubleshooting and stuff like that.
I don't think like early on we established a lot of connections with iGEM teams. It was mostly just going off of whatever connections the previous teams had made, and also through like the mentorship program. So we have a mentor from ISEF Tirupati. It's pretty close to our like instant, not close, but like in a different state. But yes, and I think that that was the only means. We didn't have symposiums or whatever. And most of what we knew about the iGEM teams, I would say, was through social media. So say Instagram or Twitter or whatever. Yeah. But it's great that you guys had a symposium. Like, it would be nice to sort of ideate together, wouldn't it?
Yeah, something that I think really helped Michigan iGEM, just to give, maybe you guys an idea, because we're definitely focusing on next year starting, like reaching out to teams earlier and really focusing on getting feedback on our project at a more early stage. And something that we implemented, this season that we hadn't the previous seasons, was we like basically wrote up our proposal for our project. So everything that we were planning on doing, even down to like specific aims of our experimentation. So we got fairly detailed. We developed a proposal and submitted that to like a separate, like science competition, just so we could get like that early feedback on our project. And that, number one, really helped us as a team like finalize our plan, because we needed to present it in a format that was, understandable to somebody who was going to be reading it. And it also allowed us to get, good feedback early on in the process. We did that like in February, so like pretty early in the season, and that like really helped us. And so something that like we especially were talking to Iser Mahali about was, maybe starting something like the manuscript earlier, so or using it as sort of like a project proposal booklet, so that teams can, write up at the beginning of the season, this is what we're planning on doing, this is a little bit of the science explanation behind the aims of our experimentation, and then like sending that out so that teams can basically read, provide feedback, and get feedback, sort of thing. We thought maybe something like that might be helpful. I was just like wondering if you guys had any ideas. Do you think something like that would be helpful? Do you think that a different format, maybe later in the season? We're just kind of like fielding ideas to like get more engaged, rather than, like after August or something like that, when teams are focusing on wiki page and stuff like that.
Actually, very curious about how your team have been organized, because tomorrow, not tomorrow, the next year, Tongji China May consists of a completely different set of people, and they may brainstorm their proposals from scratch.
Yeah, that's actually I'd actually really like to hear about this too. I'm curious how you guys like organize your timeline. So I'll talk like really briefly about Michigan. So we have like our team at large, and then we have people who are on, call it e-board, but basically leadership of the team. So we have people focused on, leading science experimentation. So like, deciding what gets done in lab on a particular day, and sort of that sort of thing in like general experimental direction. So we have those people, and we have people focused on outreach and like managing the club internally, and then we have people like for finance, and then also Emily and I, helping just organize everything at large. So that's like how our club is organized. And then we in particular, basically after iGEM, so as soon as iGEM ends, we have every member of our club basically research topics that are interesting to them, and then we quickly realize, something that could be feasible, something that's, you know, important to our local community, stuff like that. And then we sort of have project proposals. And so, you know, if everyone in our club is looking, maybe they pair up and they look at different topics, you know, maybe we end up with 5 or 6 different theoretically feasible things that we do. And then we present and we talk about it as a club and we start to plan a little bit more. And then pretty much we settle on an idea that we think we can, you know, is both interesting, meaningful, and maybe feasible. So that's how we decided our project. And we did that in the time after iGEM. So by December, we had decided our project. And so we knew it going into January, and January is when we started like outlining. We had our proposal done in February, and then we like moved forward from there. So I'm curious if any of you guys follow like a different format that you've found to be really successful, anything like that.
So I think like we are the third team from our institute. The last 2 teams have been very, very helpful for us. So we sort of had a similar thing, I guess, but it was a little later in the competition. So yeah, sorry about that. So yeah, we sort of had the same kind of thing. We had proposals ready, we had presentations ready, and then we did it to our previous teams. So they were our sort of initial mentors, and they guided us through, okay, how should an iGEM project look like versus how like a science project would look like. And so that sort of that really helped us shape our project. And then we went to professors once we thought we had a good proposal ready. Professors and I mean PhDs first, and then getting their inputs, refining project, going to professors, that sort of thing.
Does your team have like repeating members year to year, or do you get an entirely new set of people?
Absolutely no repeating members.
Oh, that's
Yeah.
interesting.
Oh, does your team have people who've already
We
been
have
in
people roll over year to year. We create like a club at the school, so we have people who, you know, join and then stay until they graduate. So I was curious if that
Oh.
was common.
I see. Oh, like I said, we're only the third team. So there have only been 2 other teams. But we, like the previous members, they just are mentors for us, or they join like the iGEM community. I mean, some of them are project heads, some of them are ambassadors, that sort of thing. They don't like they're not part of the team again. So we have other projects or whatever. They have other things going on.
I just have a final slide. Thank you guys so much for coming. This was really helpful, not only to, you know, talk about issues in health inequality that are really important to, you know, our local communities and also globally, but it was also really interesting to see the scientific explanation of your guys' project, even at a high level. And the discussion was also very nice. So thank you guys so much for coming. If you have any questions, feel free to, you know, reach out back to the email that, you know, you've been communicating with this entire time. We'll send a recap email just with the information about ICER Mahali if you're interested in that collaboration. But other than that, thank you so much for taking time out of your Sunday to come and speak here. And we hope you guys have good luck finishing your wikis, and of course, good luck at iGEM. So thank you.
Good luck, guys.
Good luck.
Thank you.
Good luck. Bye.